Okay. So just rapidly, I will introduce myself, the CBS, Doha, and then Doha will be the moderator of the session and will present Mr. Eric.
Absolutely.
Hello.
Thank you, Dr. Masmoud.
So you are welcome. So I'm Sabar Masmoudi, professor at the Center of Biotechnology of SPACS, and I am the head of the Laboratory of Molecular and Cellular Science. So thank you for being so many to participate to this conference. We had certainly more requests for participation, but we were limited by the capacity offered by Google Meet. And on behalf of all the participants, I would like to thank Mr. Eric Haas, founder at Ionic Cytometry Solutions, for agreeing to present this conference about mass cytometry. I will leave, as I said, his presentation to Dr. Doha Dasi. Doha, that I would also to thank for having thought of organizing this conference and sharing it with her Tunisian colleagues. Dr. Dasi, who first carried out her studies in Tunisia and later she joined Japanese and American universities. Already she has an excellent career with a lot of merits. I would also want to thank Hela who coordinated the contact with Doha and for the organization. Just please let me share rapidly 2-3 slides to present rapidly the CBS. Thank you. Okay, thank you. So the CBS is one of the most relevant research centers specializing in biotechnology in Tunisia. The center developed research in all fields of biotechnology, industrial, food, plant, and health. So in 2020, CBS is organized in 6 laboratories and 5 facility units. Among these facility units, the analytical services. Then this unit has already a flow cytometry equipment used by most laboratories of CBS and other laboratories in the region of SPACS for different applications. Concerning the laboratory of molecular and cellular screening, we are already working in the field of health biotechnologies, and our activities organized around the 3 topics. The first project is interested in the identification, repositioning, and valorization of biomolecules for therapy of complex diseases. The laboratory has cell lines, microbial facility for cloning, but in addition, we have an in vivo screening system in zebrafish. The second project is related to genomics and diagnostics. And the third, more recent project is interested in the digital valorization of genomic data of Tunisian patient, but also for healthy individuals. So now I will give the speech to Doha to present Mr. Eric and moderate the session.
Good morning, everyone. Can you guys hear me?
It's for you.
Thank you very much, Dr. Masmoudi, and everyone who joined the presentation. Thank you for the coordination and making this seminar happen. I'm so excited to be here today to have the conference today with Eric and everyone from CBS and from Tunisia. So as you know, CYTOF is a very important technology that we would love to learn and to implement in our research. I will start first by introducing Eric. So Eric is the CEO and founder of Ionic Cytometry Solution Company, and he is a Boston University graduate. Actually, he is going to be Boston University graduate in a Master of Engineering in Biomedical Engineering. And he was at Dana-Farber Cancer Institute, Harvard Medical School as a CYTOF core supervisor. In his free time, he enjoys roasting his own coffee, weightlifting, and reading 2 to 4 books at any time given. I will give a brief introduction about myself. I'm currently a postdoc in neurology department at the Icahn School of Medicine at Mount Sinai in New York. Of course, I'm a product of the Tunisian University and specifically University of Sfax. I graduated from ENIS, from ISBS, and from the Tsukuba University in Japan. I did my PhD there, and also I moved around several universities in US before coming to New York. I enjoy exercising, volunteering, baking, and reading. So the CYTOF technology is actually a very impressive technique that, as I have mentioned earlier, it has a lot of advantages over the normal flow cytometry that we are used to use in our labs. So the mass cytometry is a next generation flow cytometry platform with several technological advances that offer advantages over fluorescence-based flow cytometry when highly parametric analysis are required. Most notably, mass cytometry does not rely on detection of fluorescence, which requires compensation for spillover into adjacent channels, instead through the use of antibodies coupled to metal isotopes. Mass cytometry can detect direct isotope peaks without significant overlap, thus ameliorating the need for compensation. So in the case of the, the CYTOF, we won't need the compensation anymore, which is great, and it saves our time, actually. In addition, more channels are available. So the primary limiting factor is the chemistry required to conjugate the metals to the antibodies with high efficiency. Currently, mass cytometry allows for detection up to 42 plus unique parameters rather than 8 to 2 parameters that comprise a typical flow cytometry panel. Several papers actually with or from big journals have mentioned and used the CYTOF technology. It has been implemented to study the mechanisms, the techniques, the screening, the diagnostics, and I would recommend these papers that have been published by another presentation given by Stanford and CytoBank. As I mentioned, the very big merit of the CYTOF over the FACS analysis is that we remove the bad guy cells or the cells that show up in the panel as requiring the compensation step. So also another advantage of the mass cytometry is it helps us to analyze several parameters with no compensation, a huge amount of parameters at the same time. So we don't need to worry about looking at like transcription factors or cytokines or growth factors, phosphatase activity, assay to to several biological pathways, and it helps us to make a lot of conclusions about the signaling pathway that we can study. You can still do the permeabilization fixation step and do the staining as a normal flow cytometry. Next slide. So now I will leave Eric to discuss about introduction to CYTOF experimental method. Thank you. Please go ahead, Eric.
Thank you, Dr. Dasi.
Okay.
Thank you, everybody, for the opportunity to speak to you today. Let me know if my microphone gets choppy.
Eric, excuse me, Eric.
Can you hear me?
Yeah, just please, it's going to be more slowly.
Okay, 1 second.
Okay, thank you.
How about now?
Yeah.
Better? Okay. Thank you, Dr. Dasi, for the introduction and context. And thank you, everybody, for the opportunity to come and talk to you today. So like Julia said, I used to run the tour at Dana-Farber and really fell in love with CYTOF and the technology. I think it's a very important tool that can be used to help further research in cancer, immunology, allergy, anything that the immune system has a role in, CYTOF can be useful. So let's jump in. So what is CYTOF? So you might hear me say CYTOF or mass cytometry. They're interchangeable. They're the same term, really. CYTOF stands for cytometry by time of flight. So this is, as Joe said, you can see many more markers. It's a newer technique. The first paper describing the technology came out in 2009 from Toronto, but it's starting to gain a lot of traction. And again, it's similar to flow cytometry, but it uses isotopically enriched metal reporters instead of fluorescent proteins. Here's how CYTOF works, basically. In the top left, in A, you have your cell, which has the antibodies conjugated to the metal isotopes. So the little spheres here represent the metals. These are resuspended in a single cell solution, and they are then passed through in B, the nebulizer, which is part of the instrument that generates fine droplets so that you ideally have a single cell in each droplet. From there, they go and get injected into the torch, which leads into the plasma. So again, this is based on inductively coupled plasma mass spec. So this plasma is generated by a radio frequency caused by this coil that you see here, and it is about 60 megahertz, and it's heating argon gas to about 9,000 degrees Celsius. So it's very hot, and it instantly ionizes all the ions and particles and everything in the cell. So at the point of the torch, the cell stops being a cell in terms of our eyes, and it becomes an ion cloud. And so now from C to D, this ion cloud is flying through the system, and you see here you have positively charged ions. Those are your metal ions that used to be attached to your cell and are now in this cloud. They go through D, which is the quadrupole, which is essentially a big filter that is filtering out all the organic atoms, any low mass atoms, or any really high mass atoms. So what you end up with after D is just a cloud of the heavy metal ions that are in the range from 75 to about 215 Daltons. And CYTOF is able to see discrete peaks between each Dalton in that range. So once they go through the quadrupole, they then go into E, which is the time of flight chamber. And this essentially is detecting how fast each individual ion goes through the system. So depending on how fast it goes, the system can detect which metal that is. So the heavier metals are going to go slower and the lighter metals go faster and will hit the detector sooner. So it then goes, and F again is showing that it can see discrete Dalton peaks. And then once you have a set of ions that are together, it's called a push. Each push is a slice, a 13 nanosecond slice of this cloud. And as it slices, it gets pushed into the detection chamber and then is detected. Once you have 10 pushes in a row that are of similar composition, the system starts to record it as an event, as you can see in G. So cell 1, that would be your first event. Let's say that's 10 pushes in a row. We have a cell, potentially. Sometimes there can be contaminants and debris and everything, just like with flow. So not everything is going to be a live, intact single cell, but that's how it works. And then it exports the data as an FCS3 file. So it's similar files to flow cytometry. So you might be wondering who's using CYTOF and why, for what purposes. On the right here, we have the breakdown of the different categories that CYTOF is used for, that papers have been published. This is from Fluidigm, the company that makes the technology. This is from their website. So you can see it's heavy, like I said before, in immune system function detection and immunophenotyping, immuno-oncology, which coming from Dana-Farber, that was a big category there. And it's used all over the world. Obviously, I want to help grow it and get it into more places throughout the world, but some of the notable institutions that currently use CYTOF to a high degree are Stanford University, MD Anderson in Texas, Dana-Farber, University of Zurich, several institutes in France have them, all over Europe, Francis Crick Institute, many universities throughout the world are using this. And again, you can just see the breakdown of the fields that are employing this technology. And like I said, it's growing, it's an emerging field. It started coming out in 2009, and then it's slowly been ramping up until about 2016, and then it turns almost exponential and has been adopted by many more people and doesn't really look to be trending down. And along with the publications ramping up, the number of clinical trials using CYTOF has also ramped up. So there are currently, this figure is from January of 2020, which there were about 75 ongoing or finished trials that were using CYTOF. Now the figure is over 100 as of November 2020. A lot of trials this summer were started using Fluidigm sells a MaxPar Direct kit, which is a basic immunophenotyping panel that allows you to identify 37 different cell subsets. And several trials were using this kit in looking at SARS-CoV-2 patients and looking at their immune responses to try and figure out what the difference between critical and non-critical patients are. So again, just some basic principles before I dive into the meat of the presentation about how to run a good CYTOF experiment. First, again, they're metal-tagged antibodies. So basically, the way to conjugate them is you take an isotopically pure metal. It has to be over 99.99% pure. Obviously, some ways of purifying different metals and elements are not going to get that. So you have to take that into account if you're going to use those. But you take the metal isotopes, you conjugate it to a polymer that then is conjugated to a reduced purified antibody, and it binds to the thiol groups in the antibody backbone. all cells are fixed prior to acquisition, so it does kind of lend itself to working with samples like SARS-CoV-2 and other infectious diseases, just since the cells are not going to be viable when you're actually running them. It's a little bit safer. It's slow, like Dua said, but the data you get with CYTOF is so robust. I've been working with data, and you have your project experiment that you're looking for, and you know like what sorts of cells you want to see, but there's so much data that you could spend days and days and days, months looking at the data, really. There's no, like Dua said, there's no compensation required because there's no overlap between the metals. I'll touch on that a little more later. That's why there's an asterisk there, because there can be some spillover, but it's not nearly as much as flow, and it's not the same kind of spillover. And then again, there's no autofluorescence, so there's no real heavy metals naturally found in biological tissue. So you don't have to worry about, that would be CYTOF's equivalent of autofluorescence, but there's no heavy metals, so you don't have to worry about that. And one great thing about CYTOF too is that you can look at surface proteins, intracellular proteins, and cytokines all in one panel, and can look at up to 50 markers in one panel, which is very useful. Instead of running 3 flow panels or 4 flow panels, you can run 1 with CYTOF. Are there any questions on the basis of CYTOF and the kind of the background before we get into the next part?
Please, everyone, feel free to ask and do not feel shy.
Anything on how it works?
Yes. Dr. Eric?
Yes.
So thank you for your presentation already, but I have one question about the metal-tagged antibodies. So do you find any problem with the metal-tagged antibodies that have been conjugated in the lab? So I think that might fail to be detected properly. Do you find such a problem?
Sorry, could you repeat the question? I had trouble hearing you.
Okay, sorry. Again, I'm talking about the metal-tagged antibodies. Do you hear me?
Yes.
So you don't find any problem with the metal-tagged antibodies that you have, that you are already conjugating in the lab? Because if you do so, you might have some problems to be detected properly by the instrument. I think we have the same problem with the attuned cytometer. I don't know about the CYTOF one.
No. So yeah, that's a good question. There's really 2 kind of ways of going about getting these metal-tagged antibodies. One is, again, Fluidigm, the company that makes it, has a pretty big catalog of pre-conjugated antibodies that are pre-validated, and those work really well in my experience for the most part, depending on your sample, obviously. And then you can conjugate them yourself in lab, and it's really not that difficult. It's a fairly straightforward process. It just is a little laborious, but again, those, as long as you follow the protocol well, they end up working usually just as well as the commercially conjugated antibodies too.
Absolutely. Thank you so much. Thank you.
You're welcome.
Eric, we have a question from the chat box.
Okay.
It's Dr. Riyad, who is from Faculty of Science of GAFSA. He's asking us, is there some specific sample preparation?
Great question too. Yes, there are specific details that are different than flow cytometry, but in general, the workflow is very similar. There's just a few more washing steps involved, just because of the chance for there to be free metals in the solution that could contaminate the machine and contaminate your sample. So like flow, you're just looking at really cells since you're gating on forward scatter and side scatter. So you can kind of exclude if there's just free antibody in the solution. But in CYTOF, if there's free antibody in the solution, it could overwhelm the detector, and that could be problematic. So yeah, there's a few more washes, and there's a specific DNA stain that I'll show later that is used. Since in CYTOF there is no forward or side scatter, we use a DNA intercalator, which actually goes into the cells. You fix and perm the cells at the end of the process, incubate them with this DNA intercalator, and that informs you what your cells are. That's one of the main differences between the two.
Yeah, thank you.
All right, no problem. Yeah, feel free to post them in the chat or ask more questions. Any risk of contaminating?
Risk of contaminating the cells prior acquisition. I mean, how does, for example, the media interfere with the results?
That's a great question. I'm actually going to cover that later, so I'll get into that and answer that when I get to the slides. That's one thing I was going to touch on, but that's a great question. There can be risk of contaminating the cells before acquisition, definitely. That's one thing that is part of the optimization process, is making sure that your processing does not have that. Okay, so we've talked about this already, the difference between CYTOF and flow, and the fact that CYTOF allows you to see discrete isotopic peaks, whereas flow, obviously most of you might be familiar with this, you have the spectral overlap, and it can cause some problems. So here's what I call it, the building blocks to success for a CYTOF experiment. First, experimental design. That's where it all starts. It's like with any scientific process, it all starts in the design phase, and part of that is the experimental design. Then you get into panel design, very important. Then into the protocol optimization, which we were just touching on a little bit, panel titration optimization. Then you get into the actual experimental acquisition, and that really, if you do the first four components well and do them correctly, the experimental acquisition almost runs itself. So obviously there's some errors that can come up, but if you take care of planning it out well, the experiments should go fairly smoothly. The data analysis, that's a can of worms that a lot of people in CYTOF can have issues with, just because of the depth of the data and the scale of having 50 markers times 100,000 plus cells can be a lot of information to deal with. And then obviously you get into your results and conclusions, and then your follow-up experiments. So first, experimental design. So it's like with any experiment, what questions do you want to address with CYTOF? And is CYTOF really the right application for what you're trying to do? And as Dua said earlier, in my experience, if you're 10 to 12 or less markers, then flow is typically going to be the better choice if you have an experienced core, or if you're experienced yourself and know kind of what you're doing with the higher parameter flow experiments. It can be cheaper, it can be less time consuming, but it's really a fine line between 10 to 12 markers. And then next, what steps do you need to be carried out? So I kind of touched on them on the previous slide. Those are the main steps, but there might be some more nuanced details that you need to think about when you're setting up your experiment. Like, what controls are you going to use, and how should you process the samples and store them? Where are they coming from? So one thing that I've seen is a lot of times people will be running samples on cells that were frozen 10 to 20 years ago, and a lot of times those don't necessarily work out. You don't know who was freezing them and how exactly they froze them. So those can present some challenges, but you can do it. You can get some information. It just might not be the best and the cleanest. And then again, what experimental conditions are you going to use? Are you going to look at treated versus untreated? Are you going to look at different time courses over a therapeutic trial, like in your project 1? Are you going to look at stimulated versus unstimulated conditions and look at different T-cell responses? And what controls are you needed? CYTOF, a lot of people do it without using spike-in reference samples, but I'm trying to change that. I think they're very important to use so you have an internal control when you're staining. Because you're pipetting potentially 50 different antibodies at a time, and you might be staining on different days, if you use a spike-in reference sample, you can see if you have any technical errors in your staining. So I think it's an important tool to use, and actually BioLegend recently came out this year with a thing called their heavy Vary cells. I don't know if any of you have worked with Vary cells before, but they're lyophilized PBMCs. So if you're working with PBMCs, BioLegend has a pretty easy to use spike-in reference ready to go off the shelf. And then ultimately, these experiments are about what story is being told. What are you trying to convey? What are you looking for in your experiment and your project? What is your objective? And then how can CYTOF help you reach that objective?
Eric, we have a great question from SCSI, from Faculty of Medicine of Tunis. She's wondering, can we study the tumor infiltrating lymphocyte TILs with the CYTOF, or the immunohistochemistry HTC will suffice, will be enough, like to look at it?
Yeah, definitely you can look at TILs with CYTOF. That's a big application field, especially at Dana-Farber is looking at the TILs and some of the tumor immune microenvironment. Absolutely. Those are great projects. They take more nuance with the sample prep, obviously, because you might not have that many set of those cell subsets in the samples, but CYTOF is a great tool for that, because with CYTOF you can look at very small amounts of cells and get a robust amount of information. So yeah.
I remember I have been doing that at Dana-Farber by the flow cytometry. I wonder if that would generate much better result using the CYTOF comparing the flow.
I know I've seen it. I remember working with your lab, and Shah had seen some better results using CYTOF than with flow. Definitely the picture more clearly.
Mm-hmm. I have one more question. How the metals correlate with the data we get, and how many metals are staining each antibody?
Good question. So the metals in the data are hand in hand. The metals, basically, when you're running the samples and you get the data exported, you're being told what metals are on that event, not necessarily what antigens are on the cell or antibodies are on the cell. So the amount of metal is directly correlated with the plots. Some plots I'll show later, but just like in H. So this is on the y-axis one element, and on the x-axis another element. And these cells here, really, this is what they are showing the intensity of the metal on each of the cells. So like these up here in the top left quadrant, those have higher intensity of metal element C and lower intensity of metal element A. So that's they're directly linked. Can be interpreted as how much of antibody or antigen is on that cell too. The number of metal atoms on each antibody can definitely vary based on which metal you're using. So there is a curve of intensity on the instrument, and there are a couple different polymers depending on where the metals fall on that curve. So like something like 89Y, which is on the low end of the spectrum and is typically used for high expression markers like CD45, that is going to have more metal atoms per antibody than higher level markers. I think it's something like there's 10 or so polymers on each antibody that get conjugated to it, and about 5 to 6 atoms per polymer, something along those lines. So you end up with anywhere from 50 to 100 atoms per antibody.
We have one
roughly.
more question from one of my professors actually at the School of Engineering. It's Dr. Hassan Haj Qasim. He's asking, what is the sensitivity of the technology to small molecule metabolites? And what is the maximum number of parameters we can measure per run? Usually we worry about the rare metabolites or rare molecules that we want to detect. So how about that in case of the CYTOF?
That's more of an emerging use in CYTOF. There hasn't been a lot of work done with metabolomics in CYTOF, but I know of at least a couple of papers that have shown that it works. I don't have any direct experience with that, but I know that there's some people that have had success looking at those. And similarly, I've worked with somebody that was looking at exosomes as well, which is more small molecule type thing. So it can be done, but it definitely takes some thinking, some more thought about the specifics of what you're looking for. Absolutely, but it can be done. And then the max number of parameters per run. So you can run, the range runs of the technical range of the instrument detection is from 75 to about 215 Daltons. So in theory, you could run about 130, 140 markers with the current setup. Now, the limiting factor there, though, is the chemistry of purifying the elements in that range of masses and then making sure that the chemistry is able to be conjugated to polymers. So that's really the limiting factor. Right now, there's about 47 to 50 commercially available isotopes to use. Not you can just buy directly from the company, but there are people, and I've played with it too, that you can use, you can buy elements and buy metal salt solutions from like Thermo Fisher or any other biotech that sells them. If they're pure, you can use those to conjugate to the polymers too. So like something like indium 115 or 113, those work really well, and those are not commercially available, but you can use them for CYTOF.
Interesting. We have one more question. Can we measure the amount of Ca2 plus calcium in cell using CYTOF? From Iman.
Now that's something that I have not seen directly, but you definitely can, and I'll show you why later. But I believe that you would be able to do it, but there could be some worries about that. One thing is that it's a lower mass ion, so it might not be easily detectable, but we can see barium. So a lot of times, I mean, I've worked with people that have high barium contamination that we thought might have actually a high degree of calcification, and he was looking at heart tissue. So you might be able to. I wouldn't rule it out.
Makes sense. Yeah, thank you. One more question, and then I will let you move forward, actually. Question from Mohammed Garifeli from CBS. How to avoid the problem of cell degradation during the analysis, and especially when it is about a weakly presented cell population? Again, it's about the rare population.
Right, so that's a fundamental. That's a fundamental thing about CYTOF 2 is that there's no way to get the cells back. So once they go through the system, they're ionized and they're no longer a cell. They go through the detector and then out into the atmosphere from the exhaust. So yeah, that is a troublesome part of CYTOF. You can't get the cells back.
So it means you can do sorting like the flow.
Right. You can sort. One workflow that I like is to use CYTOF to look for, to just kind of do a shotgun style approach and cast a wide net and look for specific populations that might be upregulated or downregulated, that might be some sort of identifier, and then go into flow and sort for that population. So they can work hand in hand. So the next step after you've thought about the experiments, and these are all great questions, thank you for asking them, I really appreciate it. The next step of the process is the panel design. So there's several considerations, and the first is obviously what antibodies do you want to use in your experiment? What are you looking for? You want to look at T cells? You want to look at B cells? What exactly do you want to see in your experiment? The next is, like I mentioned briefly, the mass sensitivity of the detector is there's a window of best sensitivity metals to use. So 155 to about 175 Daltons is where the detector has its brightest. Now, when I say there's a range of like best sensitivity, it's minor compared to some flow markers and how some flow markers show up, like FITSI versus a lower expression flow marker. The range difference is not that great in CYTOF. So you would want to put your more rare markers in this 150, 160 range, but you can get away with putting them in some on the outskirts of that range too and be fine. So that's kind of what this panel wheel over here, this is from the MaxPar panel designer. And this wheel is basically showing you how much spillover you're getting, which spillover, like I said earlier, we don't have spectral overlap in CYTOF, but there are 3 types of spillover that can occur. So the first is just M+1 abundance. So let's just say on this wheel, let's say you have 143 ND and you have something in 144 ND. Now, neodymium has the most isotopes of any element in the catalog, really. So there's going to be, you can't get these isotopes completely 100% pure. So in 143 neodymium, there's going to be a fraction of 144 neodymium. So, but the beautiful thing about CYTOF is because it's more based on physical, like hard physics, you can predict and see the amount of spill that you're expecting to get from that 143 into 144. So you can kind of control for that. You can also have isotopic impurities, which is kind of what I touched on. So in 143 neodymium, there's also going to be a fraction of 150 neodymium. So that's, again, just part of how they purify the isotopes. They can't get them 100% pure, so there's going to be some spill. And then the last one is oxidation. So oxidation is usually relatively low because the instruments are tuned every morning to be below 0.03% oxidation. So while there can be some oxidation that occurs, it's less likely, but it can happen. So what that looks like is you'll have whatever your mass of the element is plus 16. So for instance, 141 praseodymium is the most oxidative metal that is used in the system, and that plus 16 would be 157. The 157 channel is rarely used in CYTOF because of this, because the ability of 141 to oxidize. And another consideration when you're doing your panel design is the antibody availability. So when I'm designing panels for people, I try as much as possible to, depending on your needs and your budget really, I try as much as possible to get the your panel to be mostly already conjugated antibodies. It's just easier that way, and if you have the funding to do it, it's worthwhile to just buy them from Fluidigm directly. But obviously, there might not have everything you need, so you can do some custom conjugations, which we try to minimize. Like I said, they're not that difficult, but they are time consuming, and then you have to also quantify those and validate those. So it adds some more steps to the process. Not that bad again, so don't be persuaded if you see something in the catalog. If you have a marker you want to look at but you don't see it in the catalog, you can conjugate. So the next step is protocol optimization. Again, this is kind of what we were talking about with the contamination. I'll get back to that question. So part of it is finding the proper workflow so you mitigate any potential issues that could come up and it works well in your hands. Because the staining process, especially if you're doing cytokine staining or intracellular staining, it's a little bit longer of a process. So typically people will leave the samples overnight in the DNA intercalator that I was talking about earlier that we use for cell discrimination. They'll use that and leave it overnight to incubate just because of the duration of the process. So finding a workflow of what works, like say you come in at noon, you stain and then leave overnight in the DNA intercalator step and then come back and run the next day, or you start early in the morning, do the whole process in one day and run it in the evening or afternoon. You gotta find what works best and find what protocol works best for you and your samples. Some tissues can be a little more difficult to work with, obviously, to make single cell solutions. So finding the right kind of enzymes to break down and degrade into a single cell solution is important, and finding ones that are pure and not contaminated. So there's a lot of optimization that can be done for the protocol. So thinking about the conditions of your samples is important. Again, like are you going to be using samples that were frozen 10 years ago, or are you using fresh samples? That's going to change your protocol a little bit. And then how does the workflow function best in your hands? Like how comfortable do you feel with it? Because part of it, I think, that gets overlooked often is the human factor. Are you comfortable working with the samples and working with the protocol and doing the staining? Because again, it can be a lot. So it's important to practice, which is part of this protocol optimization process. The contaminants, there can definitely be room for contamination to be introduced before you run the samples. So one common source is actually rubber. Rubber can introduce barium to the samples. So if you're using a rubber stopper to grind your cells or dissociate your tissues, that can introduce some barium to the samples. Water, you need to use pure water. If you're using water, you shouldn't. Nowadays, water's kind of been phased out of the process. But if you're using agents like PBS, it needs to be calcium, magnesium free, so you don't have those contaminants. And then some different buffers, like if you have specific tissue types and you need to use different collagenases and DNases and stuff, those sometimes can have contamination too. So just if you're not sure, you can always just run some of the solution on the instrument and see if there's contaminants in that. You can also have iodine from, a lot of times, typically this comes from Ficol prep, just because there is iodine in Ficol. So if you take some of the plasma out and some of the other gradients that are in there, besides the PBMCs, sometimes they can soak up some of that iodine. So that can be problematic, because that's right in the middle of our range. And there can also just be some other heavy metal contamination. Like I didn't see this personally, but I've heard of somebody having samarium contamination because they were going outside and smoking cigarettes, and the lighters they were using had samarium in the striker. And that ended up getting on their hands and clothes, and then they went in and stained their samples, and it ended up getting in the samples. So really anything with heavy metals could get in the samples. I've seen tin in samples from just dirty lab spaces. So you can definitely introduce contaminants.
Wow, this tells how much the technique is really sensitive to detect them. Eric, we have two questions. One is
Okay.
from Hussein Bouasle from a university in Algeria. I'm glad to have our colleagues in Algeria here. She's saying there is any specific condition which must be taken and in respect to the synthesis of samples and antibody process or in their storage.
That's going to be really sample specific and experiment specific, but in terms of the overall protocol for staining, most of it is done at room temperature. The DNA intercalation incubation is done at 4 degrees Celsius. So those are really the main things. And again, depending on how you've frozen the samples, a lot of times they work best being frozen at 10% DMSO, which works pretty well. And then thawing them, it works well to have media and some kind of protein in the thawing buffer. When you're thawing, that's done best in a water bath. And then again, the specifics of the conjugation are pretty detailed and relatively straightforward. There's a lot of incubation in 37 degrees, and then the antibodies are stored in a 4 degrees Celsius fridge, just in some antibody stabilizing solution. The samples, if you're running fresh blood, you can store them in the fridge for a couple of days, I guess, if you're, depending on your level. It's best to either to stain your samples fresh from isolating the PBMCs or whatever your tissue type, or having them frozen and then thawing and staining. Some people have seen success with staining samples and then freezing them and then running them later. That can work too. And antibody cocktails can also be frozen and saved for later too.
We have two more questions. You can let me know if you want to have them now or we can delay them later. So one question is from Asia Said again. Thank you, Asia, for asking and being proactive and interactive with us. So her question is, for this cytometry, we detect fluorescence, and for the CYTOF, we detect what on our target? I think she means the off target of the technique, basically.
So you're detecting the metal, essentially. It's directly detecting the elements that are conjugated to the antibody. The antibody is bound to the antigen of interest, whatever it may be. So CD3, maybe it's going into the CD3. It sees that, and that metal is on, due to its antibody binding, that metal is now bound to the cell, and then it goes through the system, and we see these elements coming through. So you're detecting elements instead of fluorescence.
One more question from Sir Hassan Haj Qasim. I have a strange question. I'm thinking about to compare whole gene expression using RNA-seq at RNA level, including untranslated RNA, and a whole proteome expression using CYTOF. Which one will be more expensive in terms of cost? Or it's better to combine both technologies, like RNA-seq result and CYTOF result?
Great question. Yeah, I think combining technologies is always a good thing if you can do it. RNA-seq is probably going to be a little bit more expensive, just based on the number of cells analyzed. But again, I think doing both, that's a great approach. That's a wonderful idea to look at gene expression and protein expression using those two. That's a common thing that I've seen, or it's starting to gain some traction, is using multi-technologies for sure. So you can definitely do that. I think it's best to combine both if that's what you're interested in. Because obviously just because something is expressed or just because something is transcribed and translated doesn't mean it's going to be expressed on the cells. So it's important to look at both. Yeah, I would say if you're going to look at RNA expression, you should definitely look at protein expression too.
Makes sense. Nice.
All right, moving forward here. great questions. Keep them coming. Let's hold off until we finish the slides for the rest, though. But feel free to put them in the chat or whatever, and we'll get to them at the end. So next step in the process is your panel titration and optimization. Titration is absolutely necessary before. Just jump right in and not titrate, but doing a proper titration is going to save so much hassle and time later on in your process that it's worth taking your time here and making sure you're doing it accurately and finding the optimal staining concentrations for your samples. There's a couple different strategies for determining that optimal concentration. And one is that you just split your panel into 3 groups. One is your high expression markers, one's your medium expression markers, and one the low expression markers. And you first titrate your high expression markers, then your mid, then your low. And you do this in basically a serial dilution and just see what your best titration is. Determining best titration is basically looking at how much spill are you getting into other channels, your signal to noise in there, how much spill are you getting in other channels, and can you see that marker well? It's essentially the balance that you're looking for. Then you can also run every other channel and just split your panel into 2 separate titration panels and run every other channel together. Then another way is just run only channels that aren't capable of spilling into each other. So like I was talking about the neodymium isotopes earlier, you could split it up into several panels that are not going to spill into each other as predicted by your panel design. So let's say you want to look at 142 ND plus 142 gadolinium. That would work. You can do that. And then another one that really is probably the absolute best way to titrate, but is probably, it's a little more time consuming and isn't necessarily always that practical to do, is to stain each marker individually and titrate each marker. Not a lot of people do it because it can be expensive and time consuming. And if you design the titration, I favor the first one, splitting the panel into 3 groups. If you do that well, you probably don't need to split it up into single metals, but you can do single metal titrations as well. And that's going to give you a very clear picture of where that marker might be spilling into and what level you need for your titration. So next is you've done all this setup, you've gotten your titration done, your experiment is working well, your protocol is working well. Now you're ready to run. So to do that, part of it's establishing your routine for your staining days, like we talked about in the protocol optimization, and then just looking and seeing how the staining worked as you're doing the process, which if you use a spike-in reference sample, it can help inform this and quantify this rather than just looking at it. To an untrained eye, you might not be able to tell how it's running, so it's important to have some sort of control so you can really quantify it. And then just troubleshooting any small issues that might come up, like clogging or contamination or potentially poor staining. Maybe there's an instrument issue because it's the high technology, it's still a piece of a machine that can be prone to having errors or breaking down. But where I come from, the person I trained is doing very well, Noah at Dana-Farber. We don't run the instrument if it doesn't pass tuning. So we would rather just shift your time or put it on another instrument than run on a suboptimal instrument. So here, this is what is actually seen when you're running samples. This is called the rain plot. And essentially what you have here is a cell. And the way we can tell that, the first place I look is over here on the right. You see 191 IR and 193 IR. This is your DNA intercalator. So this is natural abundance iridium that just goes in and binds up any nucleic acid really inside the cell because you fixed and permed it for that step. So that's how we're able to distinguish our cells. And you can see here, like one thing I'm looking for is, do I see a lot of streakiness in the background? So if there's if there's excess antibody or metal in the solution, you'll have basically just a black line in one of these samples. So that's not good. You don't want that. But this looks really good. This DNA looks good, nice and discreet, pretty clean, and you can kind of see, it's a little bit hard to read at first, but the markers are up along the top axis here. So you can see this cell that's going through is CD14 positive. It's got HLA-DR and some CD11C. So it's some sort of monocyte macrophage type cell, right? You can kind of look at that. CD45, you can tell over here, so we know it's an immune cell. So that actually would be the second step after looking at the DNA. Is it CD45 positive? Yes. But again, this plot is just to check and see how it's running. It's not used for data analysis at all. It's just a troubleshooting thing and something for the instrument operators to be able to check and ensure the sample's running well, or there's no contaminants. So in this sample, there actually is a contaminant, but it's not too bad. If you see, look over on the left, there's a 127I, which is iodine. So again, these samples were prepped with phycol. So you have some cell-specific iodine here. So these cells sucked up iodine from the phycol prep and now have it just in there, probably as all. But as long as it's cell-specific like this, it's not a big issue. Again, if it's a black streak, that's an issue, because that can damage the detector on the instrument. And if it has enough contamination, it can be at the point where the instrument actually is just getting ions constantly and can't distinguish your cell from the ions that stream that's coming in. And again, this part pretty much takes care of itself if you do the other ones well. Next is data analysis. Like I mentioned earlier, data analysis can be a kind of a sticky spot for some people, just because, or a lot of people really do in CYTOF, just because of, it's really more the fear factor of there being a lot of information here. It's not, once you get it and you just practice with it, it's not easy, I would say, but once you understand it, it's not too bad. It's not that scary. Don't let the mass of information detract from the ability of using CYTOF for your experiment. So this is a chart that I made just kind of demonstrating what my high dimensional data analysis service offers. So the first kind of step that I like to do, first two steps really, are quality control and just troubleshooting the samples, again, like in the inter-experiment acquisitions. How is the staining looking? Is it working well? Can we see the markers that we expect to be seeing in this particular sample or not? Or what are we seeing? Is there any contamination? And a very important part of CYTOF is cleanup. So you want to clean your samples. There's several parameters that are exported. I won't get into the weeds of that now, just because it's a little more convoluted, but there's like an 8-step cleanup process that you do. And you start with all your events, and you distill it down to just your live intact single cells. Then from there, you can assign metadata depending on your platform or what information you want, like treatment 1, 2, 3. Then you can do some traditional gating, which I still like to use sometimes, it lets you see the data better and get into it. And a lot of people are familiar with that from Flow. So it's definitely still used in CYTOF. Obviously, you're not going to use it to find all the information you can, but you can find some good filtering through gating. Another important aspect is dimensionality reduction. So that is what you've probably heard of TISNI by now. That is a big, huge topic in CYTOF and a lot of high parameter data, is using these dimensionality reduction algorithms that take this multi-dimensional space, like 50-dimensional space, and condense it to a 2D plot. that allows you to visualize kind of the distances between different cells. So like cells will come together and unlike cells will repel each other. And then the next step that I usually use is some form of unsupervised clustering. That's going to actually essentially gate for you, but it's going to use different clustering algorithms that are using things like distance metrics and looking at the intensity of the markers that you put into the test and determining what's different between different cells based on those markers. And then obviously you get into your statistical tests and your figure generation. And there's so many arrows on here is because it's all connected. It's not necessarily a direct linear path. You could go straight through one path and then realize when you get to the statistical test that you need to go back and do some more clustering. You need to redo your clustering, or maybe you're in the figures and you need to gate for something. So it's all tied together. And here's just a few examples of what you can do with these techniques. So like I said, dimensionality reduction is very important for visualization. And I emphasize here visualization because it's not a clustering algorithm. So you see these plots below. These are t-SNE plots looking at PBMCs, and showing in the middle is the CD45 expression, colored on a heat map statistic. And on the right and left, you have CD4 and CD8, respectively. So you can kind of see where these markers are being expressed. And this is nice for visualization, really, but it's hard to quantify from just this plot. So that's where the machine learning clustering algorithms come into play. And they can actually quantify the differences between the cells you see here. And usually they'll line up pretty well with t-SNE. So like if you were to cluster this and you had a CD4 T cell cluster here, it would show up here. Again, gating can be used, traditional gating, and this analysis can be performed in really any kind of software that's capable of analyzing FCS files. I personally like Flojo and Omic, which Omic is a newer software that's come out on online software. It's really powerful. These figures are actually created using Omic. And then next, what results do you have from your experiment? You've done the data analysis, now what? What conclusions can you draw, and how are they supporting your hypothesis or not? I think it's important. A lot of people in science get into, I've seen it a lot, they want the results that they're looking for. But I think it's important to not let that blind you to results that could show up that might be different than what you're looking for, but still are important or relevant. Or sometimes not. Sometimes you don't get any kind of relevant results and you have data, but more often than not with CYTOF, with the breadth and the depth of the data that you get, there's something useful in that. And again, if it's a well-designed experiment, you probably are going to get important data for your question. So just think about that when you're doing your results and conclusions. Have you met the goal? Have you met your objectives? And then again, what conclusions can you draw? Like if you're doing a clinical trial, looking at you're looking at the tumor immune microenvironment. What are those TILs doing? What are they expressing? Are they activated? Are they downregulated? Things like that. So like PD-1 therapy, I know Dua has worked with that in her previous. That's a huge hot topic, obviously, in cancer research, and it's used a lot in CYTOF. You can see cells that are being exhausted or are being upregulated. You can detect that. And then the next is follow-up. It's pretty self-explanatory. What are your next steps? You've done your experiment, you've looked at your results, you've done the data analysis, and you've probably you're starting to write your paper. So this step would really come after your paper writing and your synthesis of the information. What do you need to do next? Are there any follow-up experiments that you need to do? Like say you're doing your data analysis and you find something and you want to go investigate it more. experiment that you need to do. And then what can you take from what you learned from this experiment to inform you for the next experiment? It's important to just build, like I showed with the cycle, the building blocks. They all build on each other. So you don't discount what you did last year for what you're doing this year. It's all ties together. All right, questions. I know we have questions, so let's see here.
Yes, we have a lot of them actually.
Great.
Thank you. Thank you very much for all those who posted their questions or asked directly. So Dr. Adel is asking, CYTOF seems to be an interesting method for cellular proteomic acquisition and quite new in comparison to the traditional flow cytometry, for instance. What are the possible exclusive applications of CYTOF that can't be done using the traditional methods, or it is rather complementary?
Good question. It's complementary, but there's some that can't be done with regular flow or other cytometry methods. So, for instance, let's say you want to look at some sort of organoid, and you want to see if something is coming through the solution of the organoid and passing through a barrier of sorts. You can take that supernatant after it's passed through and run it on the CYTOF as basically it's called solution mode. So you're not looking at cells, but you can look and see what drugs or what antibodies, as long as they're bound to a metal, are in that solution. So that's one application that you couldn't really do with anything else. You can also do barcoding with CYTOF, which is difficult, if not impossible, with flow. So that's one approach that I didn't really mention here, but it is important to use if you're doing a vast number of cells or vast number of samples doing a big trial. You can do barcoding, which allows you to pool. There's a couple methods. One allows you to pool up to 20 samples in one tube. And that's something that you can't really do in anything else. And it's important for, again, keeping the quality control of your experiment nice and tight and sharp. But it is complementary. Like I said, the workflow from looking at your RNA, looking at CYTOF, the broad protein expression profile, and then you can take that, let's say you find something interesting that's different, a population of interest in your CYTOF experiment, but you don't really need 50 markers to investigate it further. You just want to look at your Tregs, your CD25 positive T cells, and you want to look at some of the functional markers on those, like a handful, five to 8 markers. You could just do flow, and you could go and sort those cells out, culture them, and then run them again on CYTOF and a bigger panel if you want. So it's definitely, there are applications that can only be done with CYTOF, but it also is very complementary to the other technologies.
Yeah, I guess if we have a lot of factors and a huge number of parameters, CYTOF is definitely the best. So like 18 plus and 12 plus. Also, Dr. Dora is asking, sorry, but I have more questions. Don't be sorry, Dora. Thank you very much for asking. First question is, so could the platform be customized for the analysis of functional markers? And her second question is, can we use Q-dots antibodies having heavy metals, probably she means, having metals cores like cadmium to be detected by the CYTOF, and is it detectable by those mentioned channels?
Also great questions. Yes, you can use it to look for functional markers. Absolutely. That's pretty straightforward. I don't have much more to say about that, but you can absolutely use functional markers. Q-dots, you can detect those. I would say it's a little tricky because you have to make sure, again, that they're not too much of the metal ions in those, and they're not contaminating the instrument and saturating, but you can use them. There's actually new isotopes as part of the commercially available catalog that are in the cadmium channels. So you can use Q-dot antibodies, absolutely.
We have one more question from one of my friends, Mona. Thanks, Mona, for asking. She's thanking you so much for the very good presentation. She says she learned many interesting things about CYTOF, although she's not sure about the field of research and what's the question? I think maybe she doesn't have a question at this time point.
Okay.
We have a question from Dr. Rawi Fakhfekh. Is it possible to practice other analysis with FlowJo, for example, of files? The question is appearing, sorry. Is it possible to practice other analysis with FlowJo, for example, or files obtained by CYTOF? The second question is, can we detect proteins with post-translational modification like phosphorylation by CYTOF? I think definitely.
Mm-hmm. Yeah, definitely you can look at phosphoproteins, absolutely. That's another good, Julia, you've worked with that, I know, in your lab has, so yeah, absolutely you can look at phosphorylated.
Mostly the activation of protein is a very good sign in the signaling pathway, so we are very interested to look at that, so definitely CYTOF can do it. The other question, I'm not sure I understood it. Is it possible to practice other analysis with FlowJo, for example, of files obtained by CYTOF? I think we can use FlowJo, as you mentioned. I guess also CytoBank and maybe FCS Express as well. Yes,
definitely. Anything you can analyze flow data on, you can analyze CYTOF data on. Some platforms are going to be a little bit better or worse, depending on what you're trying to do. Absolutely. But FlowJo is a great platform for it.
Yep. I think we have more questions. Dr. Delmi Horia is asking, could the CYTOF be used in cell sorting and selection of good stem cell from like...
No, you can't use it for sorting, unfortunately, because again, it just, once it goes into that plasma, the cell is completely destroyed and ionized. So you can't sort, unfortunately, but you detect those stem cells and tissues and quantify them and analyze them.
Dr. Adel Tkeli, what is the cost of CYTOF instrument in comparison to FACS, for example? Also the cost of consumables. I think the reagents needed and the kits and stuff.
Great question. The instrument is about, I think nowadays it's going for about 600,000, which is pretty pricey still. They used to go for 1.5 million, which is very expensive, but they're to about 600,000 now. And I saw earlier there was a question about using paraffin embedded tissue.
I think I've seen that, yeah.
Yeah, you can, but not on, so there's two technologies here. So with CYTOF, they have an imaging platform that you might have heard of called the Hyperion. That's basically a module that plugs into the CYTOF. And with that, you can look at paraffin embedded formula fixed tissues. It's a new generation IHC basically that allows you to look at similar numbers of markers as you can in regular suspension-based CYTOF. You can look at on their imaging platform. So you can look at slides and look at different tissue sections using that. And it's a similar technology, absolutely. That basically uses a laser ablation, ablates to slide, and then sends them through the CYTOF detector and quantifies that back to the tissue and recreates it. So you can do that. The cost of the reagents and consumables, that can be a little pricey too. I would say if you're looking at a full panel that you want to start up and develop, it's probably going to cost between $5,000 to $10,000 US dollars, depending on how much markers you're using and how many samples you're going to run. That would probably be about the cost for your initial experiment, I would say.
So Eric, you mentioned that we can look at the tissue slides in CYTOF, right? I found a question. She says, it's Dr. Awat Benjmaa. Can I use paraffin embedded formalin fixed tissue? So basically the slides also we can run them in CYTOF?
Right, yes, you can run them in CYTOF, but it's not on the Helios, which is the the technology I described here in the presentation, that's on their module called the Hyperion, which is, again, it's using the laser ablation, and it ablates the cells from the slide, sends them through the CYTOF. So it uses the same mechanism of detection and uses this similar metal channels, similar antibodies conjugated to metals, and then can detect instead of typical fluorescent microscopy where you can only see up to, I don't know, 10 easily markers, you can see up to 35 to 50 markers using the Hyperion imaging system.
Interesting. Wow. I had a question, but I think you already covered that. So I'm assuming that we cannot sort these cells and use them for downstream assay because they are not alive anymore. So is it correct? I think you said, yeah, it is correct. And is there a way to collect them alive? Like somehow in the CYTOF process, we can like collect them alive and run the downstream assays, like whether it is RT-PCR or another experiment?
No, there's no way to collect them after they've gone through the CYTOF. You can, I mean, one strategy would be to run half of your sample on CYTOF and then take the rest to the other downstream technologies. Definitely, but no, there's no way to collect them. That's one of the drawbacks of it for sure.
Mm-hmm. One more question. So why no autofluorescence in the CYTOF, like, and no need to compensation?
Good question. Yeah, so that's just a basis of what the instrument is detecting. So since it's looking at mass and using mass, it's basically using a mass to charge ratio to detect the ions that are coming through. So it's not looking at any sort of photonics or any sort of light emission spectrum. So that's why there's no autofluorescence, just because it's not going to be detected. So there will be, so like if you have cells that are going to be autofluorescent on a fax, you can run them on the CYTOF and it just will not be detected, but they will still be fluorescent, if that makes sense. Like they'll still be giving off autofluorescence, but as soon as they hit the plasma, it's gone and it's not going to be detected.
I see. Can we have more questions? Do you guys have any questions?
Yeah, if there's any more questions, I'm happy to take them.
I think we don't have any more questions. Thank you very much, Eric, and thank you, Dr. Masmoudi, and all the lab members. Thank you very much for having us and making this happen. We are so excited to help facilitate this small lecture. Thank you.
Yes, thank you. Thank you, everybody, for coming, and thank you.
Thank you so much for this excellent presentation. Very effective webinar. Thank you.
Thank you very much for the audience, and thank you. Please
thank you.
Start for any further questions.
I hope this first contact will be continued, and why not to establish collaboration in the future, Duha?
Absolutely, yeah. We'll be happy to, inshallah.
Thank you.
Thank you very much.
Yes, thank you, everybody. Feel free to shoot me or do an email or anything if you want more information. Happy to work with you. Thank you for the great questions. They're wonderful.
You also. Bye-bye.
Thank you so much. Bye-bye.